Yeah, thank you guys all for joining online. We had to move online today, but Erin will be visiting in 2 weeks. So that's on May 21st and 22nd. So for those excited to meet in person, I'll be sending out a schedule just to keep in mind. And before I pass this over, I'd like to go ahead and provide a bit of an introduction. I'm so excited to be welcoming Erin here today. Erin is a macroecologist in the sense of Jim Brown's original sense of thinking about problems over large expanses of space and time. And she really integrates those two scales. She's interested in the origin, maintenance, and conservation of biodiversity. And although she describes herself as a paleobiologist, I think we could equally call her an ecologist as well, because she works on problems like things like the latitudinal diversity gradient, basic questions of biogeography, using statistical approaches like species distribution models, but also evolutionary and mechanistic models. And she thinks about things like extinction selectivity and what the past can tell us about the present day. I should be saying it, not Erin, but Professor Erin Saupe is a professor at Oxford University. She went to Oxford actually from Yale, where we met each other. She was a postdoc, a Yves Donnelly postdoc at Yale under Derek Briggs. And before I hand it over, I should just say that we're really fortunate to have her speaking here today. Erin is one of our most awarded young paleontologists today. I was super delighted to recently hear that she's received our highest award in paleontology, the Schuchert Award, which has gone to the likes of people like Stephen Jay Gould and David Raup and Gary Vermeer. So with that, I'm really excited to hear what Erin has to say, and thank you guys all for coming to the seminar today. So thanks, Erin.
Oh, thanks, Shelley. I really appreciate that introduction, and thanks everyone for being here. I'm really sorry again not to be there in person today, but I will be there on the 21st and 22nd of this month. So really hope that I can speak to some of you then, and we can chat more about whatever I talk about or about your cool research as well. I said that the title of my talk will focus on regulators of extinction and diversification in deep time. And I thought that today I would tell you a bit about some of the research that I have done in this broad sort of theme, and then partitioned out by some sort of more specific themes, and give you a flavor of the type of research that I have done in recent years. All of which I, as Shelley already alluded to, try to approach through a spatial lens. And the three sort of broad topics that I'll be talking to you about today are controls on the distribution of organisms across the surface of the Earth, what controls sort of diversification over long time spans, and then what controls how species and communities respond to both abiotic and biotic changes. So the first case study I will share with you is no exception. It approaches these questions through a spatial lens. And I'll be talking about one of the largest scale and longest known patterns in ecology, and that is of the latitudinal diversity gradient. And I'm sure many of you, all of you, are very familiar with the latitudinal diversity gradient. It was first recognized over 200 years ago by Alexander von Humboldt when he described the increase in the variety of structure, grace of form, and mixture of colors towards the tropics. And ever since Alexander von Humboldt first described the diversity gradient, there have been over 100 hypotheses to explain why we see higher diversity in the tropics. And what's really exceptional about the latitudinal diversity gradient is the pervasiveness of the pattern. So we find the diversity gradient on land, like you see here for vertebrate diversity. We find the diversity gradient in the sea, like you see here for copepod diversity. And then we find the diversity gradient not only in animals, but also in plants, like you see here for vascular plants. And diversity gradients have been studied intensively in both the marine and the terrestrial realm, of course, in the present day. And work has been done to quantify diversity gradients at specific times in Earth history as well. But despite all of this work, there's still a lot of debate about the causes of latitudinal diversity gradients. And ultimately, when thinking about causal mechanisms, they boil down to, and they must invoke, differences in regional rates of speciation, extinction, local extirpation, or the movement of species into or out of regions. And these four processes really are the only ways to generate differential numbers of species across geographic space. These processes themselves can be controlled by a suite of abiotic and biotic factors like spatiotemporal climate change, species interactions, and available area and resources. But the problem with identifying these causal factors is that many of the factors that are thought to affect rates of speciation and extinction are actually collinear with latitude today. And you can see that here for precipitation and temperature. And this makes it really difficult to disentangle what factors are actually driving higher diversity at low latitudes. The fossil record, though, can come to the rescue and can provide us with times when the relationship between latitude and these hypothesized drivers of speciation, extinction, dispersal, so things like temperature or precipitation, were different from today. So times when they were not necessarily collinear with latitude. So by looking to the past, we can potentially gain insight into which factors may be key to generating latitudinal diversity gradients. Not only that, but the fossil record can also provide us insight on whether a modern style latitudinal diversity gradient has been consistently present over long time scales. And if it hasn't been present consistently over long time scales, what does this then tell us about those potential drivers of the gradient? So although the fossil record holds all of this potential, it hasn't really been possible to ask this type of question before because of issues of spatial and temporal resolution of much of our fossil record. But work in my lab has recently generated a new high spatial and temporal resolution data set for planktonic foraminifera called Triton. As many of you are very, very familiar, planktonic foraminifera are single-celled protists that live in the oceans. They build a hard calcite shell called a test, and they can live at a variety of depths across the world's oceans. And this new database is now the largest data set of any fossil clade to date and contains more than 500,000 records over the last 65 million years. And what we did in Triton, which makes it really great for these ecological studies across space and through time, is we ensured a unified set of taxonomic concepts. We converted all of the ages to the same geological timescale, and we rotated all of our latitude and longitude coordinates to the same paleocoordinate rotation system, so that we have a consistent understanding of where species were living deeper in time. So using this new database, this Triton, we constructed latitudinal diversity gradients at multiple time intervals over the last 40 million years. And I'm going to focus on results here where we constructed gradients at 2.5 million year intervals, but the results were really insensitive to spatial and temporal binning schemes. And I can talk more about that if you're interested in the questions. So for each of those time slices, we quantified diversity gradients using 5 different approaches, including approaches that use subsampling to account for spatial and temporal heterogeneity in the fossil data that we had. Even though planktonic foraminifera are amazing and have an amazing fossil record, there are still, of course, sampling differences across space and through time. So we have to account for that. And what's great is that the 5 different approaches that we used all produced congruent patterns. So again, I'm going to focus here on diversity gradients that we quantified using a percentile approach, so specifically the 75th 75th percentile of point level richness estimates. And this percentile approach is appropriate for characterizing latitudinal diversity gradients because it's actually the upper tail of diversity distributions that generate the shape of latitudinal diversity gradients. And that's true even today in the modern. So for example, at low latitudes today, you can have regions that are very depauperate in species, so very species poor. And this is nicely illustrated here on the in modern marine fishes, where you can see at low latitudes at the equator, you have very low diversity. But of course, you have much higher diversity as well. So again, the centile approach is appropriate for characterizing diversity gradients. All right, so what did this new data compilation and this new approach tell us about diversity gradients through time? So all of the methods that we used to estimate diversity gradients showed significant changes in the spatial distribution of planktonic foraminifera over this 40 million year interval in time. And we found that modern style latitudinal diversity gradients began to emerge only gradually about 34 million years ago, but remained fairly shallow until about 15 to 10 million years ago. So the colors here indicate the ages, with the red being the oldest going back to 40 million years, and then the blue the youngest. So this steepening of the diversity gradient towards the present day can also be illustrated graphically by measuring the actual slope through time, the actual gradient. And this is what I'm showing you here. So we estimated slope as a linear function of richness with absolute latitude. So we collapsed down both the northern and the southern hemisphere, although we did this separately as well, which you can see. And you can see that there's a steepening of the gradient towards the present day from virtually no gradient about 40 million years ago. Okay, so the fact that the diversity gradient emerged only gradually over the last 40 million years means that we could then try to identify potential drivers of the changes that we saw. And to do this, we use two different modeling approaches. So in the first modeling approach, we modeled the relationship between richness and climate spatially within our time bins using spatial autoregressive models. And then in the second modeling approach, we modeled the relationship between change in richness and then change in those climatic variables at given locations on Earth over set intervals of time. And we looked at a number of set intervals of time, including 2.5 million years, 5 million years, 7.5 million years, 10 million years, and 12.5 million years. And then the four environmental variables that we examined included mean annual sea surface salinity, mean annual thermocline extent, mean annual mixed layer depth, and then a measure of temperature. And we considered both mean annual sea surface temperature as well as temperature variability within the water column at a specific point on Earth. So these climate data that we used came from the HAD-CM3L coupled atmosphere-ocean generalized circulation model, which was in collaboration with Paul Valdez, Dan Lund, and Alex Farnsworth at the University of Bristol. And we needed to use these climate models rather than, say, proxy data because our model framework required spatially and temporally explicit estimates of climate for each estimate of richness. And this isn't available with proxy data on this sort of large spatial and temporal scale. All right, so results from both of those two types of models, so those spatial autoregressive models, but also the models built through time, agreed. And they suggested that temperature was the only variable to exhibit a consistent and strong positive relationship with richness over time. And this is what I'm showing you here for those spatially explicit models within each of the time bins. And this relationship with richness persisted for at least the last 15 million years, perhaps longer, but the confidence intervals overlap zero further back in time. And this is true both for the mean annual sea surface temperature variable, but then also in models where we swap that out for temperature variability within the water column. So again, both models, those built across space and through time, agree that temperature and temperature variability correlate positively with richness, which is not particularly surprising. But what's interesting is that the changes that we see in the diversity gradient for planktonic foraminifera mirror the changes we see in the steepness of the latitudinal temperature gradient. So we see initial formation of the diversity gradient beginning around 34 million years ago. And this is actually coincident with this transition from warm house to cool house conditions, which is pretty cool that they are congruent. And then we see a significant steepening of the diversity gradient, coincident with a steepening of the latitudinal temperature gradient around 15 million years ago as well. And what's important, I think, or we hypothesize, is that the steepening of the latitudinal temperature gradient is associated with an increase in vertical temperature structure, so within the water column at low latitudes. And this is because colder bottom waters are being brought down from higher latitudes to the tropics. And this might mean that the emergence of the modern day latitudinal diversity gradient for planktonic foraminifera was caused by enhanced sort of thermal niche partitioning or sort of more heterogeneity in the environment at low latitudes within the water column. And this might have increased speciation at low latitudes or caused the extirpation of species and communities at high latitudes as warmer water niches were removed. And so if this were true, we would expect changes in how planktonic foraminifera partition by depth within the water column across space and then through time as well. And we can actually quantify this using a metric for evenness, which examines how species are partitioned across different habitats. So how many species are found within each habitat. And in this case, we're looking at depths within the water column. So low evenness would indicate more species are present at one water depth over another. And so for each time interval, we examined how species were distributed across 3 depths, so the mixed layer, the thermocline, and the subthermocline, with the goal of testing if assemblages exhibited more evenness of depths across latitudes when the gradient was shallower millions of years ago. And what we found was that assemblages were indeed more even across depths and across latitudes when the gradient was shallower millions of years ago. And this might imply that warmer waters at high latitudes supported a broader range of vertical temperature habitats, but that these assemblages collapsed as the high latitudes cooled. Again, makes sense. So if the warmer waters at high latitudes supported a broader range of vertical temperature habitats, we would expect higher extirpation or extinction in polar regions as those warmer water niches were removed. And then we would potentially expect higher rates of speciation at low latitudes as more niches are created when those colder bottom waters are being brought down into the tropics and creating more heterogeneity or vertical structure in the water column. So what we then wanted to do was quantify rates of speciation, extinction, extirpation, and dispersal in low and high latitude regions, which here we defined as within and exclusive of 30 degrees latitude. Of course, both elevated extinction and extirpation at high latitudes and/or elevated speciation at low latitudes would help contribute to the emergence formation of the diversity gradient. So we wanted to see which is it. So what we found, though, is that speciation was the primary contributor to the emergence of the diversity gradient over the last 40 million years. So low latitude speciation began to exceed high latitude speciation after about 30 million years ago in a consistent fashion. Extirpation, so local extinction, at high latitudes also contributed to the diversity gradient by removing some of those species at higher latitudes, but it had a smaller effect size compared to speciation. And extinction, so wholesale extinction of taxa, did not contribute to the diversity gradient because actually extinction was higher at low latitudes beginning about 20 million years ago. And then same with dispersal. So dispersal from high to low latitudes didn't contribute to the emergence of the diversity gradient, so adding species into the tropics because dispersal patterns were reversed. You have more dispersal from low to high latitudes, at least for the last 10 million years. And so these results taken together suggest the modern day latitudinal diversity gradient for planktonic foraminifera is controlled primarily by speciation at low latitudes and secondarily by extirpation at high latitudes over the last 30 to 15 million years. And maybe this is really due to sort of the enhanced heterogeneity or vertical temperature structure in the water column at low latitudes. So you have high latitude cooling beginning around this time. This high latitude cooling brought colder bottom waters to the tropics, which increased latitudinal temperature gradients, but also increased vertical temperature gradients at low latitudes. The increased vertical temperature structure within the water column at low latitudes may have enhanced speciation opportunities, niche partitioning, and we do see evidence of this in our higher speciation rates at low latitudes beginning about 30 million years ago. And what's interesting is that we also find that the tropics today are richer than the tropics were 40 to 20 million years ago. And this might potentially be due to that stronger vertical temperature structure that was weaker during those warmer time intervals, or others have hypothesized that it's also might be due to the fact that the tropics were just too warm during these really warm intervals of Earth history, say 40 million years ago. And this general framework and a hypothesis that we came up with has been suggested in the literature previously, but this is sort of the first time where we're putting all these different pieces together and testing it in a more quantitative way. So what's exciting, I think, is that this work has shown for the first time how a diversity gradient has emerged slowly over time, over an extended interval of Earth history, and it maybe provides us with a more mechanistic understanding of how biodiversity is assembled. So that's sort of a whirlwind tour through the first study I wanted to chat to you about. But in addition to determining how biodiversity is assembled spatially, I'm also interested, as I said, in the factors that regulate processes like speciation and extinction. And these, of course, then feed back to influence biogeographic patterns as well. So I'm particularly, again, interested in how differential rates of speciation and extinction might contribute to the emergence of the diversity gradient, so sticking with latitudinal diversity gradients. And in the next study I'm going to chat to you about, I used eco-evolutionary simulations to test whether climate change, so both across space and through time, could generate terrestrial latitudinal diversity gradients from no starting diversity pattern. And my hypothesis here was that the dynamics of climate change across space and through time could drive higher rates of range fragmentation at low latitudes, which would then result in speciation piling up species in the tropics. So in other words, the spatiotemporal dynamics of climate change may cause more range fragmentation at low latitudes. This then causes higher rates of allopatric speciation at low latitudes, and this might contribute to the latitudinal diversity gradient. And essentially what I wanted to know with these eco-evolutionary simulations, which many of you, I think, in the room are very familiar with, have used before, is whether I could replicate latitudinal diversity gradients using only a few species characteristics interacting with a dynamic climate over a set interval of time. So in this case, 120,000 years from no starting diversity gradient. So how did I do this? Well, I created virtual species that could move across a simulation landscape that was divided into grid cells. And these species could move based on a dispersal ability that they were assigned, and they could only occupy grid cells that were climatically suitable for them. These climatic tolerances were also assigned, but the climatic conditions of each of those grid cells would change through time depending on how the climate changed. And this, of course, would then alter or affect whether those grid cells remain suitable for the species. So in this case, the changing climate conditions were represented by a 120,000 year modeled record of terrestrial climate change from the Pleistocene to the present day. And year 0 here is 120,000 years ago. And then we're stepping forward into the present every 100 years. And each point here represents a specific point on Earth that remains static. But of course, the temperature and precipitation conditions are changing through time. And so you can see how that's then shifting the shape of these points in environmental space. So that shifting climate through time, so essentially the spatiotemporal dynamics of climate will cause different processes that we talked about previously in the study on planktonic foraminifera. So local extirpation, extinction, speciation, and species movement or dispersal across the landscape. And in our simulations, if populations remained isolated for a sufficient period of time, so if two populations get fragmented, they remain isolated, then you can have allopatric speciation, and that's illustrated here. And then extinction, wholesale extinction, can result if all of the occupied suitable habitat for a species disappeared, or if a species couldn't keep pace with changing climate based on their assigned dispersal ability. So what we found just in the simulations was that we actually could replicate the latitudinal diversity gradient using the simulation framework. And I still think that this is pretty amazing and exciting because all we did was place some initial points on the surface of the Earth, give those points a dispersal ability and a tolerance level, and then watch how those points responded to realistic estimates of how the climate has changed over the last 120,000 years. And we started from no diversity gradient, so we actually had only one sort of simulation per latitudinal band, so even diversity. So from no starting diversity gradient, you could actually produce a pattern that is fairly similar to empirical patterns that you see for birds, amphibians, and mammals. And delving into it a bit more, because this is a simulation framework, we can then actually look at what's driving this. And what becomes clear is that higher rates of speciation at low latitudes are what's contributing to the emergence of the diversity gradient in the simulation. So the spatiotemporal dynamics of climate near the equator are fragmenting ranges more there, causing that isolation, which is leading to speciation. And this is really similar to what we saw in the empirical data for planktonic foraminifera, where speciation at low latitudes, perhaps because of this environmental heterogeneity, is driving higher rates there. So because of this, we could actually look at what climatic variable was actually causing the range fragmentation and therefore the speciation. And what becomes clear is that precipitation was the driving force. It's particularly the actual spatial and then temporal dynamics of precipitation. This raises the question of, can this sort of mechanism, is precipitation, is it always so variable in this specific way such that you can extend this further back in time, right? Because obviously this is only for the last 120,000 years. And maybe the answer is yes. So there has probably been low latitude variability in precipitation throughout much of Earth history because atmospheric moisture is a strongly non-linear function of temperature, meaning any small variation in temperature is amplified by the moisture cycle. So as long as the tropics were warm, high variability in precipitation would likely to have been present. So I think potentially what's beginning to emerge from these two studies on diversity gradients is that richness may be higher at low latitudes due to elevated speciation. And this may be driven by environmental and climatic heterogeneity across space and through time. But of course, there's still a lot more work to be done. So we actually used these same simulation framework to ask about factors that drive differential rates of speciation as well. Specifically, we wanted to use this in silicio framework to ask whether ecological niche evolution, so trait evolution essentially, but specifically with regard to species tolerances, whether that spurs diversification in response to climate change. And there are contrasting expectations regarding how niche lability or stability will influence rates of diversification. So one set of ideas thinks that climatic niche lability may increase rates of diversification, by reducing extinction or elevating speciation rates. Rates of climate-driven extinction are expected to be low in species that evolve climatic tolerances rapidly, given their ability to adapt and withstand new conditions, whereas rates of speciation may be high in species that adapt quickly to local climatic conditions. So populations experiencing different climatic conditions over time may undergo divergent selection, which could lead to reproductive isolation and speciation. And greater sort of lability in the niches may also facilitate colonization of new environments, new regions, and this could create further opportunities for allopatric speciation. The other side of the debate proposes that niche conservatism might spur diversification. So when you have niche stability, that's actually going to elevate diversification. So slow rates of niche evolution or niche change might increase diversification via allopatry because it would maintain geographic isolation. So species are unable to cross climatic barriers, they're unable to hybridize, and so this would lead to increased opportunities for isolation and therefore allopatry. So the idea here is that you maintain isolation through niche conservatism, and this would increase rates of speciation and therefore spur diversification. So yeah, one of the reasons for these opposing camps is that it's actually really difficult to test this question because it crosses broad scales of space and time. And correlative data can be really difficult to disentangle because of sampling biases and difficulties with current methods. So what we wanted to do is use the same ecoevolutionary simulation framework to look at how rates of niche change might affect diversification. So the simulations are really similar to the ones that I just presented for the latitudinal diversity gradient study, but here we used a fancy new isosahedral grid. So it was working sort of with equal area and equal distance grid cells. And so that's exciting. And then, of course, we incorporated evolutionary change scenarios. So we had specifically 10 different niche change scenarios, one in which no change at all occurred, which we call the niche conservatism scenario, and then others where we allowed niche change to occur in response to how climate was changing in the area where the species was living, so adaptation, but also scenarios where niche change would occur randomly. Climatic niche change could occur over the course of the simulated species lifetime, and then daughter species for speciation events were bestowed the same niche parameters as their parents, at least initially, but then they could evolve independently. So you had change occurring within the lifetime of the species. And what we found was that compared to the niche conservatism scenario, net per capita diversification was significantly lower for all niche evolution scenarios, except for the directional niche shift and directional niche expansion at 10% change, so a lower rate of change. And then the omnidirectional expansion scenario, so you had change occurring in both directions of your niche, so high tolerances and low tolerances at that a low rate of change as well. And in those scenarios, it wasn't higher diversification, but just no significant differences, so very similar diversification rates. Net per capita diversification was higher in the niche conservatism scenario because of elevated rates of speciation. So we're getting sort of a similar story across these studies. So speciation rates were statistically elevated for niche conservatism compared to all of the other niche evolution scenarios, aside from the 3 scenarios of random change. And those 3 scenarios of random change had higher speciation rates. And then the directional niche shift scenario didn't differ significantly. So we had 3 scenarios that did actually have higher speciation. And then rates of extinction were also statistically elevated for the niche conservatism scenario, which makes sense, right? They can't adapt to these new conditions compared to the other niche evolution scenarios. So there are some exceptions there. The 3 random niche change scenarios, again, had higher extinction rates, which you would expect. But these higher extinction rates were balanced by the higher speciation rates for the niche conservatism scenario. And that's why you had higher net diversification relative to the other niche evolution scenarios. So we also found, and we're able to look at the question of broad climatic tolerances, those for the niche expansion scenarios, they did not experience higher rates of allopatric speciation in our framework. So lineages that evolved broad climatic tolerances over time didn't actually have higher rates of allopatric speciation. And this is actually contrary to this hypothesis that broad environmental tolerances might increase susceptibility to vicariance events. So coming back to this question of whether niche conservatism spurs diversification in response to climate change, here the answer seems to be yes, at least based on how we have obviously modeled in our simulation framework. So either rates are similar, so there's no sort of difference between rates, or their net diversification is higher with a more stable niche. Of course, this is only with allopatric speciation, though. So the results might change if you consider things like sympatry or even parapatry. All right. So last but not least, although I often although all of this has been focused sort of on understanding the past and using past data, but I'm really interested in using that past data, as Chelly mentioned, to better understand and contextualize what's happening today in the present, particularly with a focus on conservation. So what I've been doing is looking at trying to understand community and species responses to environmental changes and what causes some species to be most vulnerable to change, which we refer to as extinction selectivity. So extinction selectivity is essentially the fact that not all taxa have the same risk of extinction. Taxa with certain traits might be more vulnerable to extinction than other taxa. And we can examine the groups and species that have gone extinct during past intervals of time and those that did not to get a sense of the traits that make species most vulnerable to change. So things like feeding strategy, motility, lifestyle mode, etc. And past work on extinction selectivity in the marine realm, using the fossil record, has shown that species geographic range size is one of the most important predictors of extinction risk, with large geographic range species being less prone to extinction. Body size has also been found to be selective, with smaller bodied taxa going extinct more readily in the marine realm. But aside from body size and geographic range size, few other traits have actually been examined with regard to their selectivity across the Phanerozoic. And this includes potentially really important traits that might regulate how species respond to environmental changes over time. So things like realized thermal preference and realized thermal niche breadth. And it was also previously unclear whether traits intrinsic to organisms, so things like body size or thermal tolerance, are better predictors of extinction risk than external abiotic factors, like the amount of temperature change experienced by individual taxa. So we decided to actually examine the relative importance of both these intrinsic biotic traits and then extrinsic sort of environmental factors in a multivariate modeling framework, including 3 traits that haven't really been looked at before over the Phanerozoic. And those are realized thermal preference, realized thermal niche breadth, and then that amount of temperature change experienced by taxa, so within the region that a taxon lives. And our analyses were relying on data from the paleobiology database. We cleaned and we vetted the data, and I can talk about that if you're interested. And the final data set consisted of over 400,000 occurrences in about 10,000 unique marine invertebrate genera. These data, so these fossil record data for unique occurrences in space and time, were then used to generate trait information for taxa by time interval. And we did this, again, accounting for the sampling heterogeneity in the fossil record. So we needed to standardize for sampling biases, or not standardize, but account for them. So for these new traits that require spatially explicit estimates of climate conditions through time, we again relied on the HAD-CM3L coupled atmosphere-ocean climate model from colleagues in Bristol, but this time that was run over the Phanerozoic at stage level. And again, these models provide us with a means to estimate these climatic conditions across space and through time that just isn't available with proxy data at this time. And these are examples of sea surface temperature for these different time periods, and the continents here are in black. And of course, the continents look different so far back in time. And we use these sorts of models then to extract our estimates for thermal niche breadth, thermal tolerance, and magnitude of climate change. Using these trait values then, which we measured for species and genera at specific times over the Phanerozoic, we could actually then use a modeling, put it all into a modeling framework. And in this case, we used a generalized linear mixed effects model, and we included stage and family as random effects, so we can account for phylogenetic relatedness, as well as the unique sort of boundary conditions for given time periods. And what we did was looked at the relative importance of these predictors by running all combinations of these variables. And what we found is that all 5 variables were significant predictors of extinction risk. And the best supported model based on AIC was the one that was most saturated, so that included all 5 of those predictors. And this means that the new traits that we tested, those 2 intrinsic traits, absolute realized thermal preference and realized thermal niche breadth, they were able to significantly predict extinction risk. And that external abiotic factor of the magnitude of climate change experienced by taxa was also just as important as those intrinsic traits in predicting extinction risk. And based on our models, taxa with narrow realized thermal breadths of less than about 15 degrees Celsius, living predominantly in the poles or the tropics, are likely to be at greatest risk of extinction. And our results suggest that taxa previously identified as extinction resistant may still go extinct if the magnitude of climate change is great enough. And I think this has some implications for understanding how biodiversity may fare under ongoing climate change today. It doesn't really matter what traits they have, even if they have, say, a large geographic range size, they're still going to be at risk of extinction if climate change is large enough. So to end, I thought I'd just put these key points here that hopefully you can take away from what I talked about today. And I'd like to thank, of course, you for listening, for being here. Thank Chelly again for the invitation. And then thank all of these people who are really critical to the research that I presented. And it wouldn't be possible without their collaborations, and often they're leading on these projects.
Excellent. I wish we could clap as a whole audience. Thank you, Erin. With the size of the audience here, I think I will open up for questions. And if you just want to turn your camera on if possible, or just speak up when you have a question, I would say go for it. And if we get a bit of a backlog, just raise your Zoom hand and we'll go one at a time. Does anyone want to kick things off?
So maybe I can ask. So first, nice to meet you. I hope to see you in person very soon. And great talk. I had a question about extinction and the other factors that could cause extinction. Of course, what we dealt with was mainly terrestrial mammals in our study that we did recently. But I was wondering whether you could see that biotic factors could be, I mean, like competition could be also like an important driver of extinction in marine ecosystem.
Yeah, that's a great question. I mean, I think the answer is potentially, but I think it's really difficult to test over such long timescales. And also on the temporal scale that we were analyzing. Some work in my lab previously that wasn't associated with the study that I showed you was looking for signals of ecological release, especially after big mass extinction events. There's this anecdote that lots of paleontologists talk about, which is that after mass extinction events, you often have these disaster taxa that take over, and you find them, more widespread than other taxa. And it was sort of thought that maybe this is an example of ecological release, where once you get rid of much of the diversity in a region or ecosystem, then it allows a taxa to expand and take over. But in that work, and again, this is marine invertebrates, so we were looking at things like brachiopods and bivalves. We didn't find any evidence for it. So actually, this classic signal of expansion of ranges after mass extinction events or drops in diversity, it just wasn't the predominant signal. So, certainly you could see it in certain taxa. That's without question. But if you're looking across all taxa during these intervals, you don't see it. So in that sense, I would say, actually, that might be one of the best ways, looking at the dynamics of range size and co-occupancy. And in some past work that I've done, we don't see that. But, whether you have sort of more complex biotic interactions with regard to food web, et cetera, yes, I'm sure that's true, but I think it's just really difficult to disentangle on the timescales that we can typically look at.
Thanks.
Any other questions? So I can, I'll ask a question while people maybe think a little bit also. I was, for the temperature change experience being like one of the emergent things from this paper, I'm really excited to see it. I was wondering if you could get into that a little bit more, because given that you're stepping at stage level, so these are, tens of, like 5 to tens of millions of year time chunks, I'm actually surprised that it matters, because the amount of temperature change that is
Going
to an
on.
actual species on a yearly scale is like insignificant. And so I was wondering if you could talk about that a little bit more.
Yeah, I mean, I think that this is, we've chatted about this, I think, in the past, where you have this really interesting discrepancy between the amount of change that taxa or like an individual or a species experiences on, say, even a daily basis from like day to night, and then certainly like an annual basis. So then how is it that, say a 2-degree temperature change will be problematic for a species or will cause a species to shift their distributions poleward, which we're currently seeing happen today, of course. And I think it's a really interesting question, and I think it has to do with sort of more extreme events that might lead to sort of extirpation or extinction, and then potentially just passing that threshold where you're shifting if a species has a tolerance distribution, but then you're shifting the climate, you're just basically maybe making it more likely that at least large portions of where they used to live are no longer suitable for the taxa, at least in certain times of the year, for example. So maybe it has to do with sort of distributions of or how much the climate is changing and shifting the distribution of environments where the taxa lives outside the distribution of their tolerance limits for certain periods of the year.
Well, I guess what you also said earlier was if you're sort of selecting or you're filtering for species over time that are overall very conservative in their niches, you can imagine them sticking in their niche until they probabilistically go extinct, I guess. So they'll just hang out in refugia and eventually blink out, I guess, also.
Yeah, potentially. Yeah. I mean,
Yeah.
that would be really, yeah, that's really interesting actually. But and certainly like I think I was really surprised that we were actually finding any signal. So we thought, oh, like let's actually characterize the magnitude of change, temperature change within the region where the species is living, right? So more of a specific estimate. And let's see if that actually predicts patterns of extinction. And the effect size is actually quite large. And it's distinct. What's really cool is that we looked for a correlation across the variables because I thought, oh, well, this variable might be correlated with, say, like the temperature breadth or temperature preference, and they're not. So yeah, I I was pretty surprised as well. But I guess it makes sense if you're just thinking about on these long time scales, you had a lot of change. We know that there was a lot of change, and obviously that comes with a lot of other perturbations as well. So something that, of course, isn't incorporated are, the changes in oxygen and changes in pH and changes in nutrients. And so like probably it's picking up, some really complex signal from the,
that are like all correlated with
yeah.
Wilson cycles or something. Okay.
Yeah.
That makes sense. Cool. Did that give someone else a chance to think of questions? Okay. Well, if everyone is anyone? If everyone is too shy, then I can just say that we'll have a chance to meet with Erin one-on-one in 2 weeks. I'll send out a schedule to meet up. We even do like the classic group lunch on one of those days. So thank you once again, Erin, and we'll talk to you more
Yeah,
in a couple of weeks.
Thanks everyone for tuning in, and thanks, Chelly, for the invitation.